So I'm going to be talking about my work at the Human Genome Sequencing Center in the clinical lab, implementing genomic medicine projects. And I'm going to include in that the work we've been doing for all of us on our investigational device exemption with the FDA. I'm happy to say that that paper was just accepted into genome medicine, and I'm supposed to be working on the final revisions right now, but I will hopefully get them into genome medicine by Friday. So, so we have supported a number of genomic medicine projects at the Genome Center, and to do so, we've built out an infrastructure for those projects. So we started with the eMERGE3 network, where we had 2 sequencing centers. So we had our group at Baylor, and we had the Broad producing sequencing data, and then we had a network of clinical sites across the country. I think there were 12 of them. Together we did 14,500 samples there. We also work with the NIAID. We have a collaboration with Mayo on the RIGHT-10K comprehensive pharmacogenomic study, with cardiovascular clinics in the Texas Medical Center. We've been conducting the heart care study. We're part of the CSER Consortium, which is the Clinical Sequencing Exploratory Research Consortium. We collaborate with Texas Children's and with Baylor Genetics to return results on pediatric cancer. And we're part of the All of Us research program, which is seeking to sequence 1 million participants across 4 genome centers and return results directly to participants around the country. So to support these projects, we have built out an infrastructure. And I just want to start with a quick definition of what we mean by genomic medicine. So we just mean to manage disease, taking into account individual genetic variability. And so I'm going to assert that in order to tackle the problem of genomic medicine from the standpoint of the clinical lab, we need to tackle 6 different problems. And this leaves out many important problems, but really from my perspective, these are the key things that I'm thinking about. So first, we need to detect genetic variation. We need to interpret that genetic variation. Then we need to generate reports, and we need to maintain and update our existing body of reports that are out there. We need to maintain compliance with regulatory bodies. We have to provide the results to a clinician, hopefully in a way that they can get to them easily. And then we need to take action on genetic information. So we're going to walk through these one by one, and I'll give examples of the infrastructure that we've built for tackling these problems. So the first one is detecting genetic variation. We're going to start with SNPs and indels. And this story starts really at the beginning of all of us, when we were thinking ahead to the scale that we would need to operate at. And we wanted to put our pipeline on the cloud, and we wanted to look at the options that were out there and assess whether what we were doing, sort of our standard approach, which was BWA and an in-house variant caller called XAtlas, whether there are any improved options at that time. So we did a fairly extensive set of benchmarking. On the left, you can see the runtimes for Dragon were very impressive, much faster than what we had been used to. And on the right, we're summarizing a whole lot of data here. So this is across SNPs and indels on 10 samples, NIST samples with gold standard data. So we're putting a lot of stuff together here, but Dragon did quite well on this set, especially beating our previous approach. So we ended up selecting Dragon for the All of Us program. Now, straight out of the box, it was not perfect. And specifically, we noticed that there was an issue calling variants with low allele fraction. So you can see here, this is a control sample. We call it the Horizon sample. And it is specifically designed to test your pipeline's ability to detect low allele fraction variants. We noticed that Dragon was missing two variants here with 25% and just under 17% expected allele fraction out of the box. So the reason Dragon misses these is that with the standard parameters, it's optimized toward the maximum F measure. So it gives sensitivity and precision equal weight. I think, I don't know, this might be controversial, but in a clinical lab, you have an entire pipeline, your secondary confirmation pipeline, that's devoted to detecting false positive calls. And so you can actually, I would argue, tolerate a small number, I'm not saying a ton, but a small number of false positives. Whereas if you have a false negative, you really have no recourse. So we worked with the Dragon team on this problem a bit, especially because I think it's sort of a growing area of research interest, and we're increasingly understanding that these possible mosaic variants can have pretty large health implications. We wanted to make sure that the research data included them. So we worked with the Dragon team to make sure that we could detect these. And the approach that we took was to try to find a parameter set that would make the change in F measure minimal, but would allow us to recover these variants. And the key change that we put in was that Dragon penalizes variants that deviate from an expected diploid model. So 50% for heterozygous variants, 100% for homozygous variants. And it allows you to put a cap on the maximum penalty that you receive for that deviation. And so when we added that cap, we were able to recover these true positive variants. The F measure changed very minimally. It did go down very slightly though. And so this parameter set was accepted as the harmonized parameter set across all of us sites. And so we put this infrastructure on the cloud. It gives us a bunch of advantages, including can manage the environment of each job in containers, get continuous integration, which helps with deployments. We have automated data lifecycle, which I really like because I've spent a lot of my career managing data in different environments. And it just handles itself in our environment, which is wonderful. And one of my engineers, Nura, gave a wonderful talk to AWS about our pipeline. So if you would like more details, feel free to Google. That's the title of her talk. And she goes into a lot more detail about this. The high level of how it works is that we have our local environment. We push files up to the cloud where they sit in buckets. There are different lambdas that are monitoring those buckets, watching for different files to appear. And if, say, a fast queue appears, it launches specific jobs. So it will launch Dragon, it'll write back to the bucket. The CRAM appearing will launch other jobs, QC jobs, et cetera. Everything gets written back, and then we push down, we push back down to our local LIMS system at the end. And so we've really ramped up for all of us production. We've done just under 50,000 samples as of a couple of weeks ago. You can see in April, we had this sort of inflection point where we increased our rate, and it was really amazing to watch the system handle that increase in load. We really didn't have to do much. We just took some caps off of some service limits for AWS, and it was fine. So that was very exciting for me to see. Okay. So that's how we're detecting SNPs and indels. The next piece I wanted to talk about is detecting CNVs. We built a tool called Atlas CNV for detecting copy number variants. This is specifically designed for gene panels. We're also working on a whole genome version of this. So it's a coverage-based method for CNV detection, and it works by finding regions within each sample where that have higher coverage than what you would expect. So for all coverage-based methods, you need a reference. You need something to compare to. And what we found the best comparison set is actually what we call batch normalization. So these are samples that were prepared in the same library and that were sequenced together. we've had the best results. We found those are the best comparators. This relies on having a large enough samples in that batch for you to be able to have a good comparison. So that's the one caveat. But the way the Atlas CNV works is that for each exon, we calculate an average depth of coverage and we normalize taking our PKM. And then we compare in our batch at at that exon across the batch. We find the median coverage level, and then we assess for in the other samples whether there are any outliers from that median. And then we perform a QC and a confidence assessment. So the QC all works based on variability. So we can look within a sample and measure the coverage variability. If it falls outside of our expected parameters, we say this is too variable for us to call CNVs in. We call that a failure. And then we'll actually look at an exon across the sequencing batch. And if the the coverage variability at that exon across all of our samples is too variable, we may need to drop out either that entire exon or specific exons from that batch. We then calculate a confidence score, which Ted called a C-score. The intuition here is it's based on the distance that our outlier is from the distribution of coverages in the other exons. And the idea is that a larger distance from the median means a more confident CNV call. And so we compared that method to another similar method, and one of the biggest impacts we saw is a reduced false discovery rate. A lot of this comes down to the effectiveness of that C-score for picking out the confident CNV calls. We were able to apply this in eMERGE, and we were actually able to get down to single exon CNV detection, which is no small thing. CNV detection is more challenging the smaller the CNV gets. We had a 70% accuracy rate on those single CNV detections across a set of controls that we sent for MLPA confirmation. Okay. So I've given you two quick overviews of how we detect genetic variations. Now we need to move into interpreting it. And the first, which many of you I think are familiar with, first challenge in interpretation is just there are so many variants we need to filter. So if this was an example from eMERGE 3, this was a data freeze of about 10,000 samples. And just with no filtering, you have over 95 million variants. Once you filter by quality and allele frequency and mutational effect on the protein, so the obvious things, you can get down to about 10 variants per sample. And then when you apply the clinical sites reporting wishes, as well as our internal database of variants that we have reviewed in the past, you can cut it down to about 9,000, so a little less than one variant per sample to review. These go to a manual review. They're assessed for ACMG criteria for whether or not they should be included on the ultimate clinical report. And at that time, we had about 770 positives, which were about 2/3 of highly penetrant pathogenic alleles, and about 1/3 were risk variants, so low penetrance pathogenic alleles. Oops. Similarly, in the All of Us program, we have started aggregate review. So health-related return of results is not launched yet. So we're not able to look at an individual participant level before we receive consent for return of results. What we can do is look across big batches of samples. So what we do is we combine at least 300 samples into a single mega sample, and in doing so we lose the knowledge of which sample a variant came from. We then perform interpretation in that way. We also lose the zygosity information when we do this. So we're not really able to interpret genetic disease properly. But the idea is for us to be looking ahead, getting ready for health-related return of results in all of us, and to assess the resources that we'll need during all of us health-related return of results, and to give the program information on positive rates. So you can see, in this subset we had reviewed just under 40,000. We have improved our rate of review a little bit over what we had in eMERGE. This is both by tightening our filters a little bit and by building up our local variant database further. So the overall results of reviewable variants were about 4% positive. So it's a little over 2% of samples have a positive variant right now. If we break down the positives by disease, we see a lot of recessive disease, which, this will change when we get to actual patient or participant reporting. We also see breast cancer, cardiomyopathy, hypercholesterolemia, long QT, so the expected disease areas. We manage all of our variant interpretation with a system we call Neptune, which we try to apply across our clinical reporting projects. It works in a very straightforward way. We have variant data coming in, it gets filtered. If everything is already in our database or filtered out automatically, we just have an automated report that goes for generation and approval. If there are new things that need review, we go to a manual review interface, and the results of that manual review are put in the database, and then this sort of cycles so that hopefully the second time through, it is fully automated, go to report generation, manually approved, and it gets sent out to the clinical site. The data I'm showing on the right side here, this is a rolling average of the number of variants reviewed per sample. And this was sort of an experiment where we pretended we had an empty VIP database at the start of the project, and we kind of replayed our review over time. And the idea was to see, how does this curve change as we add things to our database? So you can see at the beginning, just applying our basic set of filters without being able to use our internal variant interpretations, we're reviewing, about 10, just under 10 variants per sample, which quickly drops as you review, say, the first 1,000 cases, but then we sort of plateau around 1 variant per sample. So this represents this sort of long tail of rare variation that others have commented on as well. This is the continued sort of review burden that you will have as you do these high throughput sequencing projects. So another system I wanted to talk about is answering the question, if let's say you have a whole genome, which genes do we want to focus on? And the idea is to capture phenotype from, say a clinician who can provide phenotype to the clinical geneticist, which they do through an interface. And we take advantage of the HPO graph structure to do inexact matching of terms and to come up with a list of genes who have phenotype terms that match the input phenotype terms that we got, for this patient. So we can use this to get a list of genes to prioritize in a particular sample that's really tuned to the phenotype that this particular patient has. It can change for every patient. Doesn't have to be the same for a whole project, for example. And then we can send this and potentially order a targeted exome based on the set of genes that we've identified based on these phenotypes. Another tool I'm going to talk about in the genetic variation is interpreting CNVs. So interpreting CNVs can be quite a challenge. And so one of the intuitions that we had about this is that if you imagine this setup, so the blue line is our genome, and we have some feature in that genome. In this case, it's an exon. And then let's say we have a known pathogenic CNV that we see in ClinVar, for example, and that's this dark red. Now we're trying to interpret two novel CNVs, this little one here, CNV1, and this novel CNV, CNV2. Well, the overlap of CNV2 with this known pathogenic CNV is actually quite large, but it doesn't overlap this key feature in the genome, which CNV1 overlaps almost perfectly. So the idea here is that we need to take these key features into account when we're looking at CNVs. And so that's what Cassandra CNV is designed to do. It allows you to provide a set of features that you're interested in. And sort of by default, it is ClinVar CNVs and exons in your capture or in your targeted region. And then it will provide annotations of the overlap of the new CNVs that you're seeing with those key features, along with information from some other major data sources that help for interpretation. And in the top left here, you can see just sort of an example of running it on an A12878, just without doing any annotation. You have 111 CNVs to review. But if you do this overlap, you end up with 7 to take a look at. So now we have detected and interpreted genetic variation, and we need to get it communicated to the clinician. The first step in that is to put it on a report. So this is an example of a report from heart care. We had 4 main sections. We had the highly penetrant pathogenic alleles. We had a polygenic risk score section. We had LPA, LP(a) risk alleles, and we had a pharmacogenomic section. And there's a lot of work that went into the language on here to try to make it as understandable as possible. And we had 2 sort of general sections of the report. There was a summary page, and then there was the back pages for very detailed, contain a lot of molecular detail on the findings. Like I said earlier, we use Neptune to generate these reports. The way that Neptune functions is that it pulls from all the many systems where information about a patient lies. So we have to get data from the limbs, we have to get variant curation information, we get sample data, including variant data, coverage data, QC data. We have to get data from our gene knowledge base. And then, of course, our variant database pulls in, and Neptune handles taking all of that and putting it onto a single structured data file that's acceptable to go to the EMR. And we'll talk a little bit more about that later. So then now we have this body of reports that we've issued. And the thing is that genetics is very changing rapidly. So we need to maintain and update these existing reports. So this is the problem of reanalysis. And here's 2 quick examples from the eMERGE project. So we built a system called Review. And what it does is it takes, we have a snapshot of ClinVar that we used at the time of review of variant interpretation prior to the report being issued. And then we have our internal database of variant interpretations. So when we go to do a reanalysis, we take a new snapshot of ClinVar and we look for new assertions. So things that are in the new snapshot that are not in the old snapshot that disagree with our internal variant curations. And that gives us a set of ClinVar assertions that disagree with our internal curation and are new for us to review. So for example, here we had, that process gave us 26 variants that were upgrades. So we had it internally as a VUS or benign, likely benign. Someone in ClinVar put in that it was a potentially likely pathogenic or pathogenic. So we had 26 variants in that case. They affected 29 samples. So we reviewed those variants and we ended up issuing 3 corrected reports based on that information. Similarly for downgrades, so these are cases where we had it as pathogenic in our variant database. Someone put a new assertion into ClinVar of VUS benign or likely benign. And so we had 86 variants that actually impacted 310 samples. And we had two, based on that re-review, we had two corrected reports. Another pathway by which this can happen is simply communication with the clinical site. So we had, during eMERGE, a set of variants that are VUS, but they're very, very close to being likely pathogenic. So they may have been, one ACMG subcategory away from being likely pathogenic. So we kept a list of these variants, and then we, at one point in the program, we communicated with all the clinical sites about the presence of these variants, and we said, do you have any additional phenotype information that you can share about these variants? And that process was very interesting, and it led to 4 updated reports. Typically, this happened because getting the additional phenotype information, allowed us to assign a new case versus control, to identify a new case for these variants, which put us over a threshold of cases versus controls, which allows you to assign an ACMG subcategory. And then one of these was, the phenotype was just highly specific for a disease that was known to be associated with the variant that was under review. So we'll come back to this idea a little bit later, but this communication back to the clinical site for more detailed phenotype information, given specific genetic information, I think can be a very fruitful pathway to pursue. So another problem that we have been thinking about is, so given the previous work where we're updating clinical reports, we now have multiple versions of a report in circulation. And this, typically clinicians are interacting with these reports either as a PDF that's been loaded into an EMR, or that we even had some clinical sites that were printing them out, so they had paper copies. So the we started thinking about that problem. How do you know if you have multiple versions of the report? How do you know you're looking at the most recent one? We came up with a system we called Arbor. And the way Arbor works is that every report has this QR code on it. And we maintain a ledger of reports using strong encryption, and the entries in the ledger are linked with a one-way hash, and that captures the content of the report. So then as we issue reports, we update the ledger. There's then a client API, it's like a Python script, and there's also a mobile app called Arbor Scan. And what you can do is, so you have the mobile app, you scan the QR code, Arbor Scan will connect with the clinical lab, it'll get the latest ledger, and then it can check the current report that you're looking at against the ledger. And it'll tell you whether or not you're looking at the most up-to-date report. And also, it actually can tell you whether it is in fact a report that we issued, if, for some reason there was some mix-up there. So one thing about the way this is designed is that because that ledger is strongly encrypted, it can live on this mobile device. And even after the project ends and we shut down our internal database, the mobile app still functions because it just has the final ledger for the project, and it can continue to be used to check the QR codes on these reports. Okay, so now we're issuing reports to clinicians. There's return of results activities going on. We're obviously going to get scrutiny from regulatory bodies. I think that in large part, I don't think this is controversial, that these genetic tests have been regulated as LDTs, as lab developed tests. And we've developed a bunch of these in our clinical lab, but all of Us program ended up being a little bit different because of its very large scale, the fact that results were returned directly to study participants without a clinician handling the return of results, and the fact that a participant or potentially their healthcare providers could make medical decisions based on these reports. So because of these factors, all of us leadership at the NIH consulted with the FDA, and they decided that the program does present a significant risk to study subjects, and that we would need to get an investigational device exemption, which was preferred over full approval. And this is acceptable because this is not something that's commercially marketed, but it will be undergoing significant modifications. If you have full approval, those modifications become much more challenging. So this was our timeline for doing this investigational device exemption. It was almost a 2-year process from beginning to end. We ended up sequencing over 1,300 whole genome samples. They cover 59 hereditary disease risk genes and 7 PGX genes. Every gene is represented with a control, and all the PGX alleles are represented with controls. And all of the metrics that the FDA asked for were broken down by genomic context and variant type. So we needed to show our performance in high GC regions, in repetitive regions, in our genes and in our genes of interest, and in the PGX alleles that we were going to report on. The process started with the decision that we needed to seek an IDE, and we started on this process of doing pre-submission inquiries, which are written document where we put in what are the data that we're going to present, and what data does the FDA want to see, and what guidelines do they have for the comparisons that we do. We put in a series of pre-subs that cover different aspects of the program. So the analytical validity, the return of results, the consenting process, et cetera. So these are all captured in different pre-subs. And then after submitting these, the FDA would respond to us both in writing and with a call, and we would get feedback on each of the pre-submission inquiries. We collected all of the information from these pre-subs, and we put in one big IDE submission in June 6, 2020. And then over about the next month, the FDA sent us a whole slew, I guess 52 requests for additional information, in some cases different additional data collection, clarifications, et cetera. And these typically had a turnaround time of 24 hours that they wanted. So we responded to all of these, and then and at the end of that, the FDA approved our investigational device exemption. So the current status of all of us is that the IDE is accepted. There's whole genome data flowing from the genome centers to what we call the DRC, the Data Resource Center. And we're currently implementing this just blizzard of flowcharts that cover sample flow and manifests between all the different entities that are involved in health-related return of results. Okay, so now we've generated this data and we're good from a compliance aspect. We want to make sure that the data is provided in a way that is easy for a clinician to work with. Historically, that has meant using HL7v2. There's an example in the bottom right here. It's really not a very flexible format. We did use it in heart care to push a subset of our information into the EMR, including the overall genetic finding and the Lp(a) finding. But there's a lot of material on the report that's not captured in this message and does not appear in the EMR, which makes it very hard. So in the future, it makes it very hard to search for these results if you're trying to look back over a patient's history. It also makes it very hard, on the report, we have a whole lot of explanation of what an Lp(a) finding means, as well as any, caveats to the detection. And that's all sort of lost when you go to this HL7v2 format. So really the better solution is the FHIR format. So Malai at our group and Larry Babb have been leading this effort. They have a FHIR specification that captures really all of the elements of a report. And they piloted it with Northwestern and Johns Hopkins at the end of the eMERGE program. And they were successfully able to generate a set of reports in this format and push them into these ancillary EMR systems that are designed to take in FHIR bundles. We're also looking at a new collaboration with University of Texas that's going to be using this FHIR format. The format's available on the HL7 website. It's currently in draft format, and they're seeking feedback on the format, people willing to pilot it in their studies. So now we've gone through the whole process of generating and delivering the data, and now we need to work with it and hopefully be able to take action on it. So I have a few sort of quick examples of how we've done this in specific studies that we've been part of. The first of which is the RIGHT-10K, which was a comprehensive pharmacogenomic screening. This was in collaboration with the Mayo Clinic. And it was really very comprehensive. It was 77 pharmacogenes and included a manual assessment of novel variants at over 10,000 participants. Really, the upshot here is that almost every participant, I mean, it's over 99% of participants, had at least one gene that was impacted by an allele that's thought to be have an impact on the response to different drugs. And most patients had multiple alleles. So you can see, this range of 3 and 4, most patients fall into those bins of 3 and 4. So, the upshot of this is that almost every participant would benefit at some point in their lives from having this pharmacogenomics knowledge deposited in their EMR. Of course, it depends on the drugs that the physician is going to prescribe. Another example of this is from the eMERGE study. So, overall in this very large study, we broke down results by whether they're indication-based or non-indication-based, so secondary findings, as well as whether they were site-specific. So these were genes that specific clinical sites wanted, but not necessarily every single clinical site wanted. And you can see our positive rate varies between just under 2% to a little over 4%, depending on whether there's indication-based or non-indication-based. Typically, our results were in cancer or cardiovascular, so arrhythmia, cardiomyopathy. We also saw asthma, chronic kidney disease. In non-indication-based, familiar hypercholesterolemia and hemochromatosis made up a substantial fraction of our positive findings. Non-indication based results, we had a lot of CHEK2 and ATM variants. I think these results are quite in line with what we've seen in other large screening sequencing programs, as much of eMERGE was either healthy patients or healthy participants or patients who are not ascertained for a particular disease trait. And so we're seeing pretty similar numbers in all of us research program. A specific example of this comes from our collaborators at Northwestern, where they broke down their cardiovascular related results by gene, and they split out whether there was an intervention that was carried out after the return of results. So the blue are interventions that took place after results, after they received the report. And so you can see that we had a variety of actions that were taken in the clinic based on receiving the clinical report, the genetic test, including more testing, so MRIs, ECG, stress tests, et cetera, and even different therapies, so cardiac ablation, angiography, catheterization, et cetera. So, really, this is showing the impact that genetic testing has in the clinic. potentially these actions would not have been taken without these findings. Okay, so in heart care, one of our most striking findings was in the Lp(a) risk alleles. So Lp(a) is not routinely assessed, but it's a lipid, and increased levels of it, people with increased levels of it have a 50% increased risk of coronary artery disease. It's not routinely measured, and no clinical lab sequenced the gene. It's a hard gene to sequence. It has some repetitive regions. There is therapeutic, basically just a statin, but it's a very effective therapeutic. So when we look across our heart care results, we had just over 20% who were positive for these Lp(a) risk alleles. And then when we went back and looked at that 20% of patients, 30% of them had a known, their Lp(a) level had been assessed prior to us doing the genetic test. And for those people who had been assessed, actually 100% of them had elevated Lp(a). So this is pretty striking. I think it shows that these risk alleles are doing a good job of identifying patients who should have this additional Lp(a) level testing done. So overall in heart care, we had a positive rate of 30% that breaks down between our high penetrance pathogenic alleles, polygenic risk scores, and Lp(a) findings, and different combinations of those groups. So overall, we had an 8% positive rate with path and likely path samples. We had 20% with Lp(a) positive, and we had 50% with some sort of positive PGX finding. And this is the breakdown by gene. The gene that we had the most findings in was LDLR, which is not unexpected because a number of the clinics that were sending us patients were lipid clinics. We had a variety of genes here. There are 158 genes on the panel, and 21 of them ended up with a positive finding. So here's a specific example where heart care got us to the right diagnosis and helped with management. So this is one of David Murdoch's patients. It was a young woman with connective tissue symptoms, hypermobility, etc. And she had a dilated aorta. And she ended up having a pathogenic variant in TGF-beta2, which is consistent with Loeys-Dietz syndrome. So a lot of her features sort of looked like Marfan, but Loeys-Dietz is managed in a different way from Marfan. Chiefly, surgery is recommended sooner with Marfan, and the place where you measure the dilations in the aorta can be a little bit different. So because of the genetic results, David was able to make a very specific recommendation. So she had been receiving chiropractic adjustments, which he instructed her to stop. The aorta needed to be monitored every 6 months. You know, to consider repair at a certain dilation. Should start on beta blockers, avoid some other drugs, fluoroquinolones, and her family should have cascade testing, especially her first-degree relatives. And of course, there are pregnancy risks with this disease, and so counseling was suggested. All right, and one final use case for genetic data that I wanted to walk through comes from All of Us, and a potentially very powerful platform for discovery will be launching soon, and this is the All of Us Researcher Workbench. So right now, they have a pre-production environment that we're able to work in that we're really alpha testing right now. And I'm working on a demonstration project where we're working with the data, assessing the readiness of the platform for outside researchers to use. And we're also sort of looking across the All of Us data set to understand a bit what we have in here. So the goal of this demonstration project is to look at the variant frequency of previously known pathogenic alleles and break it down by ancestry subpopulation. So, so that's what we've done. We used the VIP database that I mentioned earlier in the talk to do this. We're focusing on the 59 genes from AMP and ACMG guidelines for return of secondary findings. Overall, we're seeing that 2.5% of participants have one of these previously curated pathogenic or likely pathogenic variants. It breaks down by ancestry group with some little under 2%, others a little over 3%, everything sort of in that range. And I should note that these groups are very different sizes right now in this, what we call the alpha 2 release. So if I put error bars on this, which I need to do, some of these would be very wide error bars and others would be much smaller. The European group is the largest group by quite a bit. And one other note is we have not assessed compound heterozygous variants in this set yet. So I just want to close with a schematic here. This is just a generalized workflow that we think of when we do clinical reporting. It includes, I think, a lot of the entities that we've talked about today and these interconnections. And I just want to close with the idea that I think this arrow here in the middle, which is really the communication between the clinician and the clinical geneticist, where detailed phenotype information is communicated back. I think this area is a little anemic, especially in our system, maybe better elsewhere. But I think that this is a really fruitful area to work. We've seen some indications that increasing the bandwidth and the sophistication of this communication can actually help with the interpretation of the results. And thinking of this as a feedback cycle, I think is beneficial for the whole process. So if you have ideas about that and you want to talk about it or anything else in this talk, feel free to email me. I would like to thank all of the many people who contributed to the work that I have put in here. I tried to put people on their slides, but I'm sure I missed a lot. And I am happy to take any questions that you have.
Thanks so much. I think Aditi Sharma, you have a question. Would you like to ask it? Then I could shoot now. Liang, I can't remember how to.
Yeah, I can.
Good.
Let me just. So I think you should be able to talk now, Aditi. Just unmute yourself.
Oh, sorry about that. Dr. Venner, it was a beautiful presentation. Thank you for that. My question to you is, how is the clinician alerted? You spoke about the QR codes. So how are they alerted if there is an update or downgrade in the report? Do they have to actively keep checking that, or do they get any sort of email or message if there is an update in the report?
So we update the study coordinators whenever we issue a new set of reports. We do not update individual clinicians. Okay. That's a really good point. I don't know if they want updates every time a patient of theirs has an additional report. We sort of relied on the study coordinators for that. The idea with Arbor was that if you were looking at a specific report and you had a question about whether it was the latest one, you could just do that double check.
Right. Okay. Thank you very much.
Sure.
I had a question. It looked like a pretty smooth launch and infrastructure build, but if you could do it all over again, are there major areas that you would have changed or built differently? And yeah, any regrets?
Yeah. So was this for all of us?
Yes.
We had one really exciting couple of weeks that I need to put in a slide for. But basically what happens is there is a feature on AWS called auto scaling, where AWS will give you more computers depending on how big your to-do list is. And there was a bug in actually some Illumina code that caused AWS to give us instances, give us computers that were useless. So what would happen was that we would have a big to-do list. So AWS would say, oh, you need more computers, and it'd give us more, but they were useless and they couldn't take on more jobs. So then it would repeat. AWS would look at our to-do list and say, oh my gosh, you have this huge to-do list, you need more instances. And it was stochastic actually. So we would get a couple useful ones and a couple useless ones. And it would just spiral out of control up to our service limit, the sort of positive feedback loop. So we had it was very expensive because you're paying for these instances to just sit there and do nothing. And everyone involved was stumped. Illumina was stumped. We had late night calls with 7 different Amazon engineers trying to figure this out. And we finally did figure it out. Amazon, to their credit, actually did refund us for all the money we paid for the useless instances they assigned. So I guess what we would change is I would make sure that by default the monitoring of what we call idle instances is not very strong out of the box. So I'd make sure we had very robust idle monitoring of idle instances, and that would have saved us quite a bit of stress.
Cool, thanks.
Sure.
Shira, I'm gonna unmute you if I can.
Thanks. Okay, so I was just wondering, I guess I kind of had 2 embedded questions. One was, did you look at any of the structural variants that come with the Dragon pipeline? And could you talk a little bit about using AWS versus using like a local implementation of Dragon?
Sure. So all of us is not returning structural variants yet, although it's on the roadmap, and we have a work group that is looking at it and has sort of defined some parameters. So we have looked at their CNV caller and also Manta, and we're not all the way through the process yet. We'll be combining them in some way and setting some parameters around what we take. So I guess the answer is yes, we've looked at it. We haven't gotten too far with it. So the single biggest thing with doing a cloud-based infrastructure that's great for us is that the workload is very bursty. So the instruments, we're doing thousands and thousands, like 6,000 samples a month. The instruments run for a couple of days, then they unload all the data on you at once. And then the lab is waiting to get metrics back so they can do all their QC and stuff. So the faster that we can get all that processed and turned back to the lab, the better the whole process works. So using the cloud, we can just get 150 dragons when we need them, when the instruments finish writing, get through all of our data, and then we release all of those for use by other Amazon customers when we're done with it. As opposed to, if we were local, we wouldn't have 150, we might have 10 or whatever, and then we would have like a big queue at all times. And I think the wait time that the lab would see for the metrics that they're interested in would be a lot longer.
Makes sense. Thank you.